Alright guys, hope you're studying for the test. At the end of this video, I'm going to kind of play a review of transcription and translation, totally optional. If you want to watch it, if you feel pretty comfortable on those two things, then go ahead and stop it. But the first couple minutes are going to be mainly based on the different RNA molecules. The first one is mRNA molecule. mRNA, an important thing is it's found in prokaryotes and eukaryotes. It's also found in the nucleus and the cytoplasm. And you can see the function there. tRNA is also in prokaryotes and eukaryotes. It is a cytoplasm only RNA molecule, and same with rRNA. RNA is also cytoplasm only. Now basically these three you will have to know on the test, but you should know their function, you should know exactly what they do. Nothing too revolutionary there. The next one is primary transcript. Obviously that's the precursor from all mRNA, RNA, and tRNA. Once again, that is found only inside the nucleus, and it is only found in eukaryotes. snRNA, also small nuclear RNA, same thing. It is a eukaryotic only RNA molecule. It's found only in the nucleus, and one of the things that it says there is plays a structural role in the catalytic role in spliceosomes. Basically, it's a processing for pre-mRNA molecule. Also, it plays a role in the processing of pre-rRNA and pre-tRNA. SRP RNA is also basically it's a part of the signal recognizing particle of the molecule. It's only found in eukaryotes as well. The SNOR RNA molecule is a eukaryotic and nucleus only, so it's in the eukaryotes and nucleus only, and it's basically its processing and the assembling of the rRNA molecule is its main function. The siRNA and miRNA are two interesting ones. Number one, they inhibit the translation of the mRNA molecule. Both these two were pretty recently discovered. They were only discovered back in 1998, so a pretty recent discovery. They're eukaryote only and found only in the cytoplasm. The SI, small interfering, actually triggers the breakdown of the RNA molecule and other types of molecules as well. So it breaks down other types of RNA molecules when they're no longer needed. Look at these a little more directly. siRNA, its origins come from mRNA, transpondence, or a virus. So it can be caused by a virus. And this is how you can see how the effect of particular traits can be, protein development of proteins can actually be from a virus. And basically what it does is it does cause the mRNA to break down, and therefore it can affect the ability of the mRNA to be actually translated into proteins and amino acids. miRNA actually comes from RNA that's transcribed from a distinct gene or area. So mRNA has to come from the living organism, whereas siRNA can come from a virus. And it basically triggers the breakdown of mRNA, and it can actually inhibit translation. Why is RNA able to do all this stuff? Number one, it can form hydrogen bonds to other nucleotides, which is kind of a key part of this. It also can assume a three-dimensional shape, and has functional groups that allow it to act as a catalyst, which is that idea that not only proteins can act as catalysts, but also ribozymes or RNA can act as a catalyst as well. We talked a lot about this stuff already, but just so if you want to keep filling your notes or see this, the difference between prokaryotic and eukaryotic in the process. Remember, eukaryotes do have that nuclear envelope that separates them, so you do need to take that pre-mRNA or the primary transcript and also allow it to be modified in order to kind of continue along its way. Once again, here kind of just shows the process. Noticing with the DNA on the bacteria, it can just kind of happen simultaneously. As transcription is occurring, so is translation. There are several types of mutations that are available: substitution, insertion, and deletion. The ones that are more damaging are going to be these insertion deletion mutations. You'd record this for the for your homework. Just to realize how these actually occur. Obviously, the earliest ones, the substitutions, may not actually cause a problem, especially if it does happen in an intron region, or another way of substitution may not affect it is if it still codes for the same codon or has the same amino acid that it codes for. We call that a silent mutation. Insertion deletions are typically a lot worse, and it causes a frame shift mutation. If we have enough of a mutation that a 3 nucleotide are either inserted or deleted at one time, it'll actually get us back in the right reading frame. So here we see a deletion mutation from the original DNA and RNA, causing a completely different, from that point on, all of the RNAs are going to be different. Sometimes it makes more sense if you look at this, not in actual the DNA RNA terms, but in like the English language. So here we see all the different types. The normal gene, the sentence says, "As the man saw the dog hit and then there's an end. Notice there's nonsense stuff at the very end. And what ends up happening is, in the first one, a point mutation, it changes dog to dot. And in deletion, it shifts the frame down. In that first case, what it ends up doing is deletes the entire, doesn't cause a frame shift. Then there's an insertion where 3 letters are inserted. The last one is, notice it's the frame shift, how from that point on no longer makes sense, and from that point it doesn't stop necessarily at the stop codon as we kind of would want. There's no stop codon down there. Once again, just kind of showing these different mutations. I'll let this sit on here for a little bit to watch. There are things that can cause mutations. Obviously, they can be chemical. There can be stuff that creates differences. Some of them are not too harmful. One of these reasons is that why twins look different. DNA methylation can actually occur, cause slight differences between twins and the expression of proteins. That's why their DNA, when they're older in life, is technically a little bit different. It's the protein the same, but the expression can be different. Alright, and then I put this diagram at the very end because I think it's pretty important for you to know and know the different parts. You might have to identify, what this thing is up here, this entire material here, these different locations, the E, the P, and the A, this molecule right there, one of these, the bond that actually forms right there between those. Those are all things I could ask you on the test. So just be prepared for that. There may be a fill in the blank section with an image that looks very similar to that. If you got transcription and translation done, go ahead and stop it here. I'm going to continue on talking a little bit of a review between those two processes. Just for the test, I promise you there will be a PowerPoint or a fill in the blank section that I have all of the different types of RNAs that I covered in this video, and I will have columns. I'll have all the names on the left. I'll have whether eukaryotes or prokaryotes. I'll have their location, whether it's a nucleus or cytoplasm, and I'll have their function. And what I'll do is I'll just leave parts of it empty. So I'll say mRNA, and you might have to, and I'll put in there prokaryotes and eukaryotes, and then you got to fill the rest. And then for other types, I will, I might leave. I might tell you it's in eukaryotes and it's found in the cytoplasm, and its function is to inhibit the actual making of the mRNA molecule, and you need to tell me that was miRNA. So that would be a section of the test. This is going to be a section of the test. I will just have a drawing of DNA replication, and you got to just label everything that needs to be in there. That'll be on there, and then also some fill in the blank. So if you feel comfortable in that, go ahead and stop this. If not, I'm going to go for about another 5 minutes on transcription, translation, tell you a couple other things, and call it good. Alright, when we look at transcription, what ends up happening is, and I'll kind of blow this up and I'll move it out of the way in a second, you can kind of see where this is occurring. You do have that promoter region. When initiation occurs, that's the first step of transcription. There's going to be like a promoter recognition, and what's going to happen is the RNA is actually going to come from, create a formation, what they call a transcript bubble, where the molecule itself, as it opens up, it's going to kind of bend up around. And at this, here's your Tata box, and you're going to have locations where it's going to have to match. In eukaryotes, the Tata box determines the start point for transcription. It's usually 25 to 30 base pairs upstream from the actual location of transcription. In this, when it happens, the promoter is not actually transcribed in the process. It's just the RNA coding region. And once that kind of occurs, as you can kind of see, it moves from the 5 prime, there's the 5 to the 3. This is going to be actual what we're going to get. We're going to need to have to splice it out. But you're going to actually have the terminator, and then after that, the termination site. So this is the part that's coded. All this promoter region, including the Tata box, will actually not be coded. The start of transcription is right after its location. There are, and I talked about 3 types of RNA polymerase: RNA polymerase 1, RNA polymerase 2, and RNA polymerase 3. RNA polymerase 1, its main function is for large rRNA molecules. That's what its main function. RNA polymerase 2 is for pre-mRNA, so that's pre-mRNA. It also helps make the snRNA and the snORNA. And then last is DNA polymerase 3, which makes the tRNA, the small ribosomal RNA, and some snRNAs as well. Elongation is next, in which the RNA polymerase moves along the DNA molecule depending on which one. For everything we're going to talk about is RNA polymerase 2. It'll move along the DNA, unwind it, and add new nucleotides. It always adds them one at a time, and it adds it always to the 3' end of the growing strand. Always adds it to the 3' end of the growing RNA strand. It'll eventually get to termination, and at that location, when it happens, the end of the transcription unit, the recognition of the end of it occurs. The separation of RNA from the DNA molecule actually happens. That pre-mRNA will actually can be modified. One of the things that can happen is it can actually have the introns and exons can be cut out in eukaryotes. Now this is interesting. I just was reading online today that in recent years they found that there are some introns in some types of bacteria. So it's very rare, but they have found some introns in bacteria. But for the most part, the pre-mRNA is going to splice out the introns, and then the exons can actually be spliced together in different combinations. And because of that, you can actually get a little bit different expression of the genes. 50 to 250 adenine nucleotides will be added to the 3' end, forming the poly A tail. That's where it gets its name. These nucleotides are not actually coded from DNA, but will be added on after transcription, so it's not part of that transcription process. Even though there will be some extra stuff kind of at the end. In addition to that, you will also have a 5' cap that will be added. The cap consists mainly of adding extra nucleotides to that mRNA, and they also cause a methylation to the bases as well, which also to the nucleotides as well, which modifies the actual mRNA itself. In bacteria, translation will happen right afterwards. In us, it'll happen a little bit later. Next slide, I'm going to talk about just a little bit about some of the difference of tRNA. I covered translation a lot in class today, so I'm not going to go repeat that, but I just want to say a couple things about tRNA molecule. Alright, tRNA is actually made from the DNA template. It is made in the nucleus and must travel to the cytoplasm itself. And tRNA can actually be repeated and copied. And basically, as I said, the amino acids in the cytosol will actually be new ones will be applied to the existing tRNA molecule. It's about 80 nucleotides long. It is part of the ribosome. The ribosomes themselves are in bacterial cells. There's usually about 20,000 of them, and they do have a different. You got to remember they do have a different density depending if we're looking at prokaryotes or eukaryotes. 70s, 80s. Basically, when you look at that, that S stands for Svedberg unit, SVENDBERG, and it's a unit of density. And here's just two animations that basically kind of show this process of translation that we talked about in class. Good luck on your test, and hopefully you're able to do well. And if you do have any questions, please come see me tomorrow.